Module 6, DNA, Proteins, and the Cell Cycle. DNA replication. A critical feature of DNA is that it can create an exact copy of itself. This is called DNA replication. DNA replication is the process by which two identical DNA molecules are produced from one original DNA molecule. You see, in order for a cell to divide, it has to create an exact copy of its DNA, so the new cell has a complete set of instructions in order to code for all the needed proteins. And when organisms reproduce, they have to pass on these genetic instructions to their offspring. So let's talk about what happens. In DNA replication, the two strands of the DNA double helix untwist and separate from each other. Each strand then becomes a template for making a complementary strand. You shouldn't be surprised to learn that this process is very complicated and occurs using many enzymes, but the overall concept of what happens is pretty easy to understand. First, an enzyme called helicase goes through and breaks the hydrogen bonds located between the nitrogenous base pairs. Those bonds are what hold the two DNA strands together. So the DNA will split apart, and then free nucleotides floating around the nucleus are added to the new strand, following the rules of base pairing, A to T and C to G. That means where there's an adenine nucleotide on one DNA strand, a thymine nucleotide will be added to the newly forming strand at the same spot where the adenine nucleotide is on the original strand. The same thing happens with cytosine and guanine. Now another enzyme, DNA polymerase, comes along behind the newly positioned nucleotides and forms bonds between the sugars and phosphates on the backbone. Notice how helicase and DNA polymerase enzymes have the suffix ASE, indicating that they are enzymes. Their root names help to identify what they do. Helicase breaks the hydrogen bonds holding the DNA helix together, while DNA polymerase joins the monomers together into a polymer. Now this same process happens in both directions at the same time from many sites on the split DNA strands. After the new DNA strands are made and bonded together via DNA polymerase, the two double-stranded molecules coil back up into two double helixes. That means that each newly formed DNA molecule has one original strand and one new strand. We call this semi-conservative DNA replication. We start with a double strand of DNA that splits apart, shown here as purple, and the nucleotides that are new will move in, and a new backbone is built complementary to each original strand, shown here in pink. I need to mention one other thing about chromosomes. Before DNA replicates, a chromatin strand will coil up into a single chromosome. After replication, the chromosome is made up of two strands, which are held together at their centromere right here. We can refer to each coiled piece held together here as a chromatid. So a single unduplicated chromosome exists before replication, while a single chromosome exists afterwards. But now it consists of two sister chromatids that are held together at the centromere. And the sister chromatids are the same as one another. We call them sister chromatids because they are identical, like twins, and each chromatid will end up in one of the two daughter cells after cell division. Now, most of the time in the life of a cell, DNA is in its chromatin form. The chromatin will only condense into chromosomes when the cell is ready to divide. So to sum up, chromatin is made up of long strands of clusters of DNA and proteins in the cell's nucleus, while a chromosome is those strands when they are condensed as the cell is getting ready to replicate. And a chromatid is one of the two identical copies of a replicated chromosome joined by a centromere. We can define centromere by saying it's the region where two sister chromatids are joined tightly together. DNA replication has to happen before a cell divides so that each cell, after the cell division, will have a complete DNA set. This also helps to ensure that offspring inherit genetic information from their parents during reproduction.